Good afternoon to everybody. Today we'll discuss about the vitamin-like compounds. The vitamin-like compounds are example for this one is choline, lipoic acid. Don't forget. So choline, lipoic acid, and inositol are the vitamin-like compounds are the example for the vitamin-like compounds. So what is a vitamin-like compounds? Actually, they structurally and functionally they are related to the vitamins, vitamin-like compounds, but they are nutrients. Okay. See the first one is choline. Choline is a family of water soluble. It is a component of the phospholipids. It belongs to the phospholipids. Don't forget, it is a water soluble and shows the quaternary ammonium compounds. What is meant by the quaternary ammonium compound? So quaternary ammonium compound means here it is a nitrogen having the nitrogen present centrally and having the positive charge and condensed with the or attached with the alkyl or benzyl groups. That's why it is called the quaternary ammonium compounds. So choline is a family of water soluble compound and it is shows the quaternary ammonium compounds. It is essential dietary nutrient. The chemical formula of the choline is or chemical name of the choline is trimethyl hydroxyethanolamine. What is that? Trimethyl hydroxyethanolamine is a choline chemical name. Okay, the name of the choline, chemical name of the choline is trimethyl hydroxyethanolamine. Where the synthesis takes place in the body, who are the or otherwise which one is the precursor for the synthesis of the choline. Serine is the precursor for the synthesis of the choline that is synthesized in the body. So it can be synthesized in the body. The main sources for the choline is here the serine. So it's a synthesis from the serine. So serine is a precursor for the synthesis of the choline. Okay. So for the to maintain the health, you should obtain from the choline that is in the from the diet as a choline or as a choline phospholipids. As a choline phosphates because it is a phospholipid compound. Okay, so you can obtain the for maintain the health you can obtain the choline through the diet as a choline or choline phospholipids. So choline phospholipids of here is like a phosphodiol choline is a example of the choline phospholipid. Okay, like phosphodiol choline. Okay. Now see the biosynthesis of the phosphodiol choline. See here it is the actually I told you this is the compound of the phospholipid. Okay, it belongs to the compound of the phospholipid. So for the synthesis of the phospholipids, we require the phosphoric acid. See here it is phosphoric acid. But for the synthesis of the phosphodiol choline, require the 1,2-diacylglycerol. So 1,2-diacyl. So the 1,2-diacylglycerol is formed from the phosphoric acid. No, okay. This phosphoric acid involved in synthesis of phospholipids. So conversion of the phosphoric acid to 1,2-diacylglycerol. So that means phosphoric acid then undergoes the hydrogenation, produces the 1,2-diacylglycerol. Okay. So this reaction is catalyzed by the phosphorylated phosphohydrolase. Phosphorylated phosphohydrolase is catalyzed this reaction for the conversion of the phosphoric acid to the 1,2-diacylglycerol. Okay. Now see the how phosphodiol choline is formed. Okay. Choline when condensed with the ATP produces the phosphocholine. This reaction is catalyzed by the kinase. Which kinase? Choline kinase. So choline when condensed with the ATP produces the phosphocholine. This reaction is catalyzed by the choline kinase. Okay. When this phosphocholine is condensed with the CTP, produce the CDP choline. When this phosphocholine condensed with the CTP, produce the CDP choline. Okay. This reaction is catalyzed by the phosphocholine cyclidyl transferase. Phosphocholine cyclidyl transferase. Okay. Whatever is formed, that is CDP choline, when combined with the 1,2-diacylglycerol. So when combined with the diacylglycerol, see here is a CDP choline. When combined with the 1,2-diacylglycerol, produces the phosphatidylcholine. Produce the phosphatidylcholine. This reaction is catalyzed by the phosphocholine diacylglycerol transferase. This reaction is catalyzed by the phosphocholine diacylglycerol transferase. This is the way of the synthesis of the phosphatidylcholine. So, for the synthesis of the phosphatidylcholine, we require the phosphoric acid. This phosphoric acid will convert to the 1,2-diacylglycerol. So, that means is phosphoric acid when undergoes the hydrolation, produce the 1,2-diacylglycerol. This reaction is catalyzed by the phosphatidate phosphohydrolase. Okay. So, here it is a choline and combined with the ATP produce the phosphocholine. This reaction is catalyzed by the kinase, which kinase? Choline kinase. Okay. This phosphocholine is when combined with the CTP produce the CDP choline. This reaction is catalyzed with phosphocholine cytidyle transferase. Cytidyle transferase. This produced CDP choline when combined with the 1,2-diacylglycerol produce the phosphatidylcholine. Produce the phosphatidylcholine. This reaction is catalyzed by the phosphocholine diacylglycerol transferase. Okay, phosphocholine diacylglycerol transferase. This is the way of the synthesis of the phosphatidylcholine. Now, see what are the biochemical functions of the choline. Actually, this choline is required for the production of acetylcholine and S-inosine. The importance of the acetylcholine is act as a neurotransmitter. S-inosine is a universal methyl donor. So, this due to the universal methyl donor, it is involved in the synthesis of the homocysteine. Already you know very well how the homocysteine, that means is methionine is how the homocysteine is synthesized. What are the functions of homocysteine? Already you know in previous class. Okay, choline is a component of the phospholipid. It is involved in the membrane structure and involved in the lipid transport the lipids. Not only the transport the lipids, prevents the accumulation of the fat in liver. So involved in the membrane structure and transport the lipids, and not only the transport the lipids and also prevents the accumulation of the fat in the liver, and it promotes the synthesis of the phospholipids and lipoproteins. It promotes the synthesis of the phospholipids and lipoproteins. See here it is, these are the phospholipids. Okay. So that's why it is involved in the promotes the synthesis of the phospholipids. Okay. So and involved in the removal of triglycerides from the liver. Remove the triglycerides from the liver. It prevents the accumulation of the fat in the liver and remove the triglycerides from the liver. So as a component of the phospholipids, it's required for the production of the acetylcholine and S-inosine. The importance of the S-inosine is the universal methyl donor for the involved in the synthesis of the homocysteine. Actually, this choline contains the 3 methyl groups. Choline contains the 3 methyl groups. That's why it is actively involved in the 1-carbon metabolism. So, due to the presence of 3 methyl groups and 1 carbon fragments, it is involved in the 1-carbon metabolism. So, choline along with the folate and vitamin B12 can affect the gene expression and metabolize the fats. Choline helps in the metabolize the fats. It is a precursor for the synthesis of the acetylcholine. Choline is a precursor for the synthesis of the acetylcholine along with the folate and vitamin B12. These are the nutrients. So, along with the other nutrients, folate and vitamin B12, they affect the gene expression and helps to metabolize the fats. Actually, choline is existing 2 forms as a water-soluble and fat-soluble molecules. Existing how many forms? 2 forms. That is a water-soluble form and fat-soluble form. But water-soluble form of the choline is when enter into the liver, so it is converted them into the lecithin. Then it is converted them into the lecithin. So water-soluble choline when enter into the liver, it is converted as the lecithin. But fat-soluble choline we can obtain from the dietary sources, from the diet. So fat-soluble choline we can obtain from the diet. So the body media absorption takes place in the body. Choline is absorbed in the GIT tract. Which type of choline? That is fat-soluble choline absorbed in the GIT tract. So choline is important bile acid. How? So because of the choline is when combined with the taurine produce the tarcocholic acid. That's why it is called the tarcocholic acid. It is bile acid. Tarcocholic acid is bile acid. So in humans, generally choline is absorbed from the intestine and transported as a free molecule in the blood and stored in the cell membrane and organelles. It is stored in the cell membrane and organelles as a phospholipids. So choline is stored in cell membranes and organelles as a phospholipids, but inside the cells it is stored as a phosphatidylcholine and glycerol phosphocholines. Glycerophosphocholines. So it is inside the cell, it is stored as a glycerophosphocholines and phosphatidylcholines. Okay. But it is choline stored in the cell membranes and organelles as a phospholipids. Stored as a phospholipids. If you see the dietary sources, that is, so availability may be choline in proteins, vegetables, and whole grains, they contain in the choline in rich amount. But for example, in proteins, that is beef, soya bean, fish, poultry, and eggs are having the choline. Vegetables, that is including broccoli, potatoes, and mushrooms are having the choline. So, and whole grains, that is quinoa, rice, and whole wheat bread, nuts, and seeds are having the choline. Okay, these are the dietary sources of the choline. See, dietary sources of the choline and depends upon normal values of the choline in different ages. As per 0 to 1 year, choline is required in males about 125 to 150 milligrams per day. In females, 125 to 150 milligrams per day. So both are having the same normal value, that is requirement value is 125 to 150 milligrams per day. That is about age between 0 to 1 year. And between the 1 to 3 years, so the male and females are required, both are required is same amount, that is 200 milligrams per day. 200 milligrams per day. Between 4 to 8 years, the male and females are required about 250 milligrams per day choline. About 9 to 13 years, so about 9 to 13 years, so between both are, that is male and female, required about 375 milligrams per day. Between the 14 to 19 years and above, that is elders, required 550 milligrams per day for male, but females are required 400 to 425 milligrams per day. So if you observe the difference between the male and female, the requirement values are, so in elders, that is 550 milligrams per day is for the required for the males. Females are 400 to 425 milligrams per day. But in pregnancy, so 450 milligrams per day is required. Lactation, duration of during the lactation, 550 milligrams per day is required. So choline is, this is the choline is required in different ages. So that is in milligrams per day. Okay, but as per NIH means is National Institute of Health. So actually choline is required. So based on the age, that is in grams per day, check it here. That is age between 1 to 8 years, 1 gram per day. Children between the age between 9 to 13 years, 2 grams per day. Between the 14 and 18 years required the 3 grams per day. Adults age above 19 and oldest is required the 3.5 grams per day. 3.5 grams per day. This is actually, so upper table shows the milligrams per day, but here it shows the grams per day. So this is grams per day, this is milligrams per day. What is the deficiency occurs? So choline, in choline. So due to the deficiency of the choline, so you can say observe the cardiovascular diseases, neurological conditions, that is Alzheimer's disease, and non-alcoholic fatty liver diseases. So deficiency leads to the Alzheimer's disease and non-alcoholic disease and cardiovascular diseases, and also is a neural tube irregularities as muscle damage. If you consumption excess, what happens? Excess means how much? That is greater than 7.5 grams per day. If you consume greater than 7.5 grams per day, so this leads to the low blood pressure, sweating, diarrhea, fish-like body odor. Why it is fish-like body odor? Because of the contained the trimethylene. Already I told you it's contained in the 3-methyl groups. Okay, that's why it is entered into the one carbon fragment. So due to the trimethylene, it shows the fish-like body odor. So when you consume more than 75 grams per day. Okay. Now see the next one is inosita. So the chemical name of the inositol is a hexahydroxycyclohexane. Hexahydroxycyclohexane. Actually, inositol shows the isomerization, that is 9 types of isomerizations. Shows the 9 types of isomers, that is in this myo-inositol or meso-inositol is most important. Out of 9 isomers, myo-inositol or meso-inositol are most important. Okay, it is having this carboxylate sugar, so it is most abundantly present in the brain and mammalian tissues. Inositol is mainly present in the brain and mammalian tissues, and the main function is mediate the cell signal transduction. And participate in the osmoregulation. Okay. Some fruit shows the highest content of the inositol. That means is nothing but is a myo-inositol. See the which type of fruits shows the myo-inositol in high concentration. That is fruits, beans, grains, and nuts shows the high concentration of the myo-inositol, but available in the form of is phytate. That means is myo-inositol is available in these, that is in fruits, nuts, beans, and grains, available in the form of phytates. Phytate means is say mix of calcium and magnesium salts and nothing but is a phytate. So that means is myo-inositol. So inositol, so also known as myo-inositol or meso-inositol. So why? Because of the its existence, it shows the 9 isomers. Out of these 9 is myo-inositol and meso-inositol. Meso-inositol most important. Okay, it's these myo-inositol is involved in the participatory osmoregulation and mediates the cell signal transduction. So it's available in this myo-inositol is available in the fruits, grains, nuts, and grains in the form of is phytate. Phytate means is mixture of calcium and magnesium salts is nothing but is called the phytates. Okay, especially orange fruit contain the highest amount of the myo-inositol. If you see the observed in the fruits, so orange fruit contain the highest amount of myo-inositol. Okay, now see the synthesis of the inositol. Actually, the synthesis of the inositol is required by glucose. So glucose is precursor for the synthesis of the inositol. Don't forget, glucose is the precursor for the synthesis of the inositol. This is the pathway for the synthesis of the inositol. So glucose will convert to glucose 6-phosphate. Already you know very well which enzyme is acted upon between these two. So that is hexokinase. So when the glucose is converted to glucose 6-phosphate, this reaction is catalyzed by the hexokinase. Okay. This glucose 6-phosphate will convert to inositol phosphate. So here it is that this reaction is catalyzed by the inositol 1-phosphate synthase. Inositol 1-phosphate synthase will be involved in the acted upon the glucose 6-phosphate and convert to the inositol 1-phosphate. Okay. This inositol 1-phosphate will convert to the inositol. By this reaction is catalyzed by the inositol monophosphate phosphatase. Inositol monophosphate phosphatase. Inositol monophosphate phosphatase. So we'll convert the inositol 1-phosphate to inositol. Okay. So how phosphodiol inositol is formed? When inositol is condensed with the CDP diacylglycerol. So when inositol condensed with the CDP diacylglycerol produces the phosphodiol inositol. Phosphodiol inositol. This reaction is catalyzed by the CDP diacylglycerol inositol phosphodiol transferase. So that means here is the enzyme is transferase enzyme. Which type of transferase? That is CDP diacylglycerol inositol phosphodiol transferase. Okay. So inositol will convert to the phosphodiol inositol when combined with the CDP diacylglycerol. Okay. See here it is a CDP diacylglycerol. See here it is a inositol. This inositol when combined with the CDP diacylglycerol, so it is converted to as a produce the phosphodiol inositol. Phosphodiol inositol. So this reaction is catalyzed by the CDP diacylglycerol inositol transferase. This reaction is catalyzed by the CDP diacylglycerol inositol transferase. So it produces the phosphodiol inositol. So for the synthesis of the phosphodiol inositol, what is CDP diacylglycerol? Okay, how can you get this CDP diacylglycerol? So CDP diacylglycerol you can obtain from the phosphatic acid. Okay. So phosphatic acid. This phosphatic acid will convert to as a 1,2-diacylglycerol and CDP-diacylglycerol. Okay. That means is phosphatic acid when it undergoes the hydrolation produce the 1,2-diacylglycerol. When condensed with the CDP produce the CDP-diacylglycerol. Okay. So this CDP-diacylglycerol when combined with the inositol produce the phosphatal inositol. Okay. So this is the way of production of or synthesis of the phospholipid inositol. This reaction is catalyzed by the CDP-diacylglycerol inositol transferase. Now see what are the biochemical functions of the inositol. So inositol is required for the synthesis of the phospholipid inositol, so which is a constituent of the cell membrane. It is a constituent of the phospholipid inositol is a constituent of the cell membrane. So it is act as a lipophilic factor along with the choline and prevents the accumulation of the fat in liver. Prevents the accumulation of the fat in liver. So the same function is that is in choline. So choline also involved in the prevention of the fat in the liver. So here it is the inositol is also involved in the prevention of fat of the liver. But it is act as a some hormone substances. So second messenger. So and at the membrane level release the calcium ions. Calcium ions, where it is present in high concentration in body. That is, heart muscle are having the high concentration of the inositol. Heart muscle are having the inositol high concentration. Okay. Phytin is hexaphosphate of inositol that is found in plants. Actually, hexaphosphate of inositol is a type of inositol hexaphosphate that is present in the plants in the form of phytin. Okay, so it prevents an absorption of the iron and calcium from the intestines. Prevents the iron and calcium from the intestine. Now see the lipoic acid. So lipoic acid also known as the alpha lipoic acid, that is ALA, alpha lipoic acid and thioptic acid. So lipoic acid also known as both, that is alpha lipoic acid and thioptic acid. Actually, don't forget lipoic acid is the sulfur containing fatty acid. Sulfur containing fatty acid. I am not saying sulfur containing amino acids. Don't confuse sulfur containing amino acid and sulfur containing fatty acids. Lipoic acid is the sulfur containing fatty acid, not a amino acid. Okay. So sulfur containing amino acids are different. So this is the sulfur containing fatty acid. So lipoic acid is a sulfur containing fatty acid. Okay. So that's why it is also called the 6,8 dithiooctonic acid. Dithiooctonic acid. What is meant by the octonic acid? The meaning of the octonic acid? That means it shows the 8 carbon fatty acids. 8 carbon fatty acids. So okay. So lipoic acid is a sulfur containing fatty acid. Okay, if the examiner asking the so what are the sulfur containing amino acids or what are the sulfur containing fatty acids, you can give the answer is don't confuse sulfur containing fatty acid is a lipoic acid. Okay, so how many sulfur atoms are present in the lipoic acid? That is 2 sulfur atoms are present in the lipoic acid. That means is lipoic acid contains the 2 sulfur atoms. So, but it is existing in the 2 forms, that is oxidized form and reduced form. Existing 2 forms, that is oxidized form and reduced form. Okay, so lipoic acid is a both, that is fat as well as a water soluble. It is a water soluble as fat soluble. Okay, so biochemical functions. See the biochemical functions. Lipoic acid is involved in the decarboxylation reaction. Involved in the decarboxylation reactions. That is example here it is the so pyruvate, when the pyruvate conversion of the pyruvate to acetyl CoA and the alpha ketoglutarate to succinyl CoA requires the lipoic acid. This is the example of the decarboxylation reaction. So biochemical functions, that is lipoic acid involved in the decarboxylation reactions. That is here it is the so when the pyruvate is converted to, that is reaction. So conversion of the pyruvate to acetyl CoA and conversion of the alpha ketoglutarate to succinyl CoA requires the lipoic acid. Now see the lipoic acid dependent enzymes. So So some of the examples are here. So lipoic acid dependent enzymes, that is pyruvate dehydrogenase complex, alpha ketoglutarate dehydrogenase, so and branched chain oxo acid dehydrogenase and acetone dehydrogenase. So acetone dehydrogenase, branched chain oxo acid dehydrogenase, alpha ketoglutarate dehydrogenase, pyruvate dehydrogenase complex depends upon the lipoic acid. So these are the enzymes or example for the lipoic acid dependent enzymes. So for example, see here it is a pyruvate dehydrogenase complex. In pyruvate dehydrogenase complex, it catalyzes the conversion of the pyruvate to acetyl CoA. It conversion the pyruvate to the acetyl CoA. Okay. Now see the alpha ketoglutarate dehydrogenase. So in this one it is catalyze the conversion of the alpha ketoglutarate to succinate with the lipoic acid. In branched chain amino acids, already you know very well branched chain amino acids. Example here it is a leucine, isoleucine, and valine are the branched chain amino acids. Okay, so in that it is involved in the decarboxylation of the keto acids in the catabolic pathway of the branched chain amino acids. Catabolic pathway of the branched chain amino acids. Okay, now see the therapeutic applications of the here it is lipoic acid. It reduces the free radicals. It is nothing but is act as a scavenger, shows the antioxidant property in brain, and otherwise it contributes to Alzheimer's disease and multiple sclerosis. Stimulates the production of glutathione and helping the recycle of the vitamin E and vitamin C. And helping in the recycle of vitamin E and vitamin C, so, and reduce the insulin resistance and brings down the plasma low density lipoproteins. Reduce the insulin resistance and decreases the plasma low density lipoproteins. And also it is useful for the prevention of the stroke and myocardial infarction. Okay, so these are the vitamin-like compounds. That is, say here it is a choline, lipoic acid, and inositol are the like vitamin-like compounds. Okay. So don't forget choline already. So once again I am reminding choline is a phosphatic compound of the phospholipids and it is essentially dietary nutrients. The name of the chemical name of the choline is trimethyl hydroxyethanolamine. The synthesis takes place in the liver from the serine, and to maintain the health it should be obtained from the diet is a phospholipids. So diet choline or a choline cholic phospholipids, that is nothing but is example phosphatidylcholine. So here it is a synthesis of the phosphatidylcholine. So for the synthesis of the phosphatidylcholine, phosphoric acid is required for the synthesis of the phosphatidylcholine. This is the initial or precursor for the synthesis of the phosphatidylcholine. So because of the phosphoric acid, it will convert to 1,2-diacylglycerol. So after formation of the CDP choline, when combined with the 1,2-diacylglycerol, it is converted to the phosphatidylcholine. Okay. So biochemical functions, it is involved in the synthesis of the produce the acetylcholine and acetylmethionine. So and is a component of the phospholipids. It means the membrane structure and lipid transport and prevent the accumulation of the fat in the liver and promote the synthesis of the phospholipids and lipoproteins and disposal or otherwise remove the triglycerides from the liver. Okay, due to it contains 3 methyl groups, it is involved in the 1-carbon metabolism. So it is along with the folate and the vitamin D can affect the gene expression and helps the metabolism of fats. So, but it is existing both water soluble and fat soluble, like is lipoic acid. Okay, so lipoic acid also exists in both water soluble and fat soluble. So water soluble choline molecule, so when they enter into the liver, it is converted to the lecithin. But fat soluble choline is absorbed in the, we can obtain from the diet and absorbed in the GIT tract. So it is important bile acid. Why? Because of the choline when combined with the taurine, it produces the tarcocholic acid. That's why it is important that the bile. Tarcocholic acid is a bile acid. Okay. So choline is stored in the cell membranes and organized as a phospholipids, but inside the cells as it is stored as a phosphatidylcholine and glycerophospholipids. Okay. The sources of the choline is protein, vegetables, and whole grains. So already you know very well this is the normal value. So that is in elders is required 3.5 grams per day. So in milligrams for elders is required in males 550 milligrams per day, females are 400 to 425 milligrams per day. So if you see the deficiency of the choline, so neurological conditions such as Alzheimer's disease, non-alcoholic fatty livers, and neural tubular irregularities and muscle damage. When you consume it in the excess, that is the excess means here is more than 7.5 grams per day, you can absorb it in the low blood pressure, sweating, diarrhea, and fish-like body water. So fish-like body water mainly due to the presence of the trimethylene, that means the 3-methyl groups. Okay, inositol is nothing but is called the hexahydroxycyclohexane. So it is available in mechanism, existent shows in 9 isomers. Out of 9 isomers, myo-inositol and meso-inositol is most important. So this participated this myo-inositol or meso-inositol involved in the osmoregulation. This is available in the fruits, beans and grains and nuts, that is in the form of this phytates. So phytates means is nothing but is a mixture of the calcium and magnesium salts. The most important thing, so fruit is orange, is actually orange contain the highest amount of the myo-inositol. The synthesis of the inositol is a precursor for glucose, is a precursor for the synthesis of the inositol. So glucose will convert to glucose 6-phosphate. Glucose 6-phosphate after the some reactions, it is converted to the inositol 1-phosphate. This inositol 1-phosphate will convert to the inositol, but it is converted to when combined with the CDP diacetyl, it is converted to the phosphoryl inositol. Okay, this is a CDP diacetyl is done. So CDP for the formation of CDP diacetyl is required the phosphoric acid. Okay, so CDP diacetyl is when combined with the inositol produce the phosphoryl inositol. This reaction is catalyzed by the CDP diacetyl glycerol inositol transferase. Okay, so CDP diacetyl and other ways are also involved in the production of the cardiolipin. We see the biochemical functions is act as a lipoprotein factor and remove the fat from the liver. So, and it is act as a second messenger for the hormones, some hormones, and release the calcium ions. Concentration, it is more concentration is present in the heart muscle, and here it is in the hexaphosphate of inositol, that is in the form of a phytin present in the plants, which prevent the absorption of the iron and calcium. Okay. Lipoic acid is also known as it. So, ALA, that means is nothing but is alpha lipoic acid and thioic acid. Okay. So, don't forget, once again, I am repeating this sulfur containing fatty acid. This is not a sulfur containing amino acid, this is sulfur containing fatty acid. So, example of the sulfur containing fatty acid is a lipoic acid. So, or otherwise is a lipoic acid, example for the sulfur containing fatty acids. So it shows the two sulfur, it's containing the 2 sulfur atoms and oxidized and reduced form. It is available in the 2 forms, that is oxidized and reduced form. Lipoic acid also fat soluble and water soluble. If you see the biochemical functions, so it is involved in the lipoic acid involved in decarboxylation reactions. The most important lipoic acid dependent enzymes are pyruvate dehydrogenase complex, alpha ketoglutarate dehydrogenase, or 2-oxoglutarate dehydrogenase, or branched chain oxoacid dehydrogenase, acetylene dehydrogenase, or these are the enzymes. So lipoic acid dependent enzymes. If you are examiner asking what are the lipoic acid dependent enzymes, you can give the immediately answer is pyruvate dehydrogenase complex and so alpha ketoglutarate dehydrogenase. You can remember at least out of 2. That is pyruvate dehydrogenase complex and alpha ketoglutarate dehydrogenase. If you see the therapeutic applications of the lipoic acid is it reduces the free radicals that mean in the brain. So that's been nothing but act as a scavenger and otherwise contribute to Alzheimer's disease and multiple sclerosis. So and stimulate the production of glutathione and helping in the recycle of vitamin E and C and reduce the insulin and brings down the plasma low density lipoproteins and useful in the prevention of stroke and myocardial infarctions. Thank you.